Hello friends, welcome to lecture 3 of week 7. So in the previous lectures we have looked at various assembly methods and the algorithms that we use to assemble the long read short read data and which helps us in achieving a draft genome assembly. So today in this lecture we will look at one of the very important method of recent times, Hi-C, which is used to construct a chromosomal level assembly. It is not complete chromosomes, but you can say it's a chromosome level assembly, which is much better than the draft assembly. So we will discuss like how it is done, what is required, and what is the outcome of it. And then we'll also look at some of the finished genomes, like the human genome, what was finished and why the earlier one was not called finished, and things like that. And some of the case studies from the draft genomes that like for example, like to which extent the draft genomes are helpful in knowing that the genomic or evolutionary information about those genomes. Okay, so basically we have already discussed it several times in this series of lectures that a draft genome is something where you have tried to assemble the genome, but still a lot of genome or a part of that genome is missing. Okay, then again, like what kind of draft? Basically it's a like really a rough draft or kind of near covering, like how much covering it has for the genome that is you are going to sequence, right? Then reference genome is something where you have mostly complete genome, but still heterochromatin and some regions which are difficult to sequence, like long range repeats or heterochromatin. Okay, and still you have information about the, like at the chromosomal level, that how the scaffolds were segregated into chromosomes. So that is kind of complete genome. For example, Arabidopsis, grape genomes. Okay, then the real complete genomes in recent times include the human genome, which has come recently, the wheat and rice, because these are of prime importance to us, and basically human genome sequencing is very important for health and other issues also. So to achieve draft to reference to complete is any level, it is an achievement, because human genome was also a draft initially and then it was completed. So it has to start with a draft, but still with the technologies and methods that you have today and the know-how, we can at least try to have a very good quality draft or like nearly complete genome also. Okay, so basically how to determine that it's a draft or complete genome, we have already seen that Busco score, LAI score, which can be used here, N50, okay, which are important indicators of the quality and contiguity of the assembly. And of course, like if you have a better quality genome, a highly contiguous genome, contiguous means like how much contigs you could connect together to form scaffold, and like how many pieces your genome is in. You should be having some prior information about the number of chromosomes also. Okay, so a better this information, the chances that you will cover all the genes or most of the genes will also be higher, that is Busco. And also the analysis that you do, because it is also errorless or basically with very less errors in terms of the sequence ambiguity or mutations or this. Okay, so that can be used for the evolutionary analysis. Okay, a good example is like human genome. So the first draft came in 2001. Okay, that was only 8x sequencing data using Sanger reads, which is 800 to 1000 base pair. Okay, it was a shotgun approach that was used, which like heavily dependent upon the breaking the DNA into pieces, making back clones and other kind of clones, then basically transformation, cloning, okay, and then basically the culturing dependent, right, to amplify the DNA for Sanger sequencing, and involved like so many institutes, money and researchers, because it was an early stage of sequencing. The resources were limited, people were not knowing that what they are going to achieve in the end. Okay, right, only fair idea was available that the human had like 23 pairs or like 24 types of chromosomes, right? So with that, basically the genome size was 2.85 billion base pairs, and that covered almost 90% of the genome. Okay, but still, and basically at that time there was also a company, Celera Genomics, and the and another group, or basically group of the scientists all over the world who were working in the academic institutes, and basically they both took some information or data from each other, and both released their own genomes, which were published in Nature and Science separately. Okay, then recently, like in 2013, the or basically the H38 assembly was issued for human. Okay, that was almost complete, and but still, like a large percent of the genome that is in heterochromatin, and gaps, right, which could not be filled up, right, those were missing. Okay, and that was a very good quality, and sufficient for analysis, but still the desire is to complete the genome from start to end, telomere to telomere sequencing, which was achieved only recently, last year, using a combination of technologies, approaches, computational approaches, right, where now we can say that a complete set of all the 24 chromosomes, the sequence of them. So basically the problem that you may face in draft genomes is that, for example, you started your research when the first draft genome came, okay, and on the human genome. Now in the second year or third year, okay, another some more data will come, right. So basically your first reference of chromosome like was at this location, say starting with like 10,000 base pair. Now in the another version of the genome, it will move to 11,000 base pair, or some variation because some portion is inserted in between, or so basically draft is always in the improvement. Okay, and the references or the in context of their position are not fixed, right. Even the gene information and those things are also not completely available, right. So they may be a good indicators of getting some of the genomic clues and do the evolution analysis also to some extent, or basically to a fair extent, but you really desire to have a complete genome, right, so that whatever you do is kind of more reliable and robust. Okay, and now using combination of Illumina, Oxford Nanotechnologies, which is ultra long reads, PacBio Hi-Fi, which really helped because these were long reads with less error rate. Otherwise ON2 reads were, or Oxford Nanopore reads were having almost 5% error, even more sometimes, right. But PacBio Hi-Fi was almost 99.9% correct, and that really helped long read and error free read to do the sequencing. Okay, and then Hi-C, which is very important, which is we are going to cover today, which helped in assessing the long distance association between the or contact regions of the chromosomes and intra and inter, and basically that helped to place the contigs in the context of chromosomes. Okay, and then finally the telomere to telomere genome came, which was 3.05 billion base pairs, and we will go into details of what was found in the final assembly, which was not present in the previous one. Okay, so basically if you see like just a recap of the previous thing, you start with a chromosome, or basically the genome is assembled is available as chromosomes. For example, human have 24 of these, like if you include X and Y as separate two, okay, or 23 pairs of chromosome. Okay, that is basically a sequence of DNA. You cannot walk along the chromosome, right, and read it and complete it and basically tell that what base pair is what. So you have to break it into pieces. So you will be extracting high molecular weight DNA or very long pieces of chromosome, we will be breaking them into pieces, right. Okay, now with you will go for sequencing, right. After sequencing, again you have got like even smaller pieces or reads, right. Okay, short read and long read. Okay, but still you do not know from where these reads are coming from, what should be combined with what. You tried all your methods and try to make contigs out of it. Okay, connecting short and long reads together. Okay, so if I call it long read, okay, connecting them together, and basically or short basically combination of long short, short long, and whatever way you could assemble them and make the contigs. Okay, then you are placing those contigs in a in the form of scaffolds using long distance information which you may get from link reads or from the long insert reads. Okay, so you got like even bigger scaffolds where you knew like, okay, this is one contig, this is one contig, but you still do not know what is it, things here, right? So basically scaffolds, contigs, you have reached to this extent. But now when you started with a genome. For example, human had 24 type of chromosome. Now where this scaffold is going to which chromosome, right? How you will map it back to that? Okay, how will you segregate these scaffolds into different chromosomes? Okay, can you really connect the scaffold that you got into like even much bigger pieces on a larger context, and also place them to the chromosome are real challenges which are required to achieve a basically complete genome or very good high quality chromosome level assembly. Okay, even if that you cannot achieve telomere to telomere at this stage. Okay, so basically you have to place it back in the context of chromosome, which is a challenge, and Hi-C method that we are going to discuss today really help in this way. Okay, so like to start with basically human genome story, that draft genome was released, and at that point of time achieving a genome before this, like this was the largest genome that could be done. Before there's the genome that were available by Drosophila or C. elegans or yeast, right? And then they started the human genome, and draft by draft they reached to the final genome assembly. Okay, and okay, so it was a important effort and involved so many people across the world, right? So this was the paper that came for the science issue, right, in year 2001. Okay, then recently, like last year, the complete genome came, which is the telomere to telomere assembly. Okay, so when we discuss about chromosome, basically telomeres are at the end of the chromosome. It has P arm, Q arm, centromere. Okay, like if you see this picture, so basically okay, centromere, the P arm, the Q arm, and the telomeres. Okay, so you have to assemble everything, the heterochromatin, euchromatin, right? So this genome covered all these parts, removed all the gaps, found more genes, and found all the repeats, right? So this was it took almost 32 years to reach to this stage. Okay, so basically typically in a genome that you start with like sample collection, right, and then do the genome and transcriptome sequencing, right? Then you do the assembly, correct the assembly for errors. Okay, then you try to merge the assemblies from long and short read together to create a hybrid assembly, again correct the errors, polish it, okay, and then report the genome statistics including the N50, BOOSCO or LAI and these kind of and then submit your data to public databases, right? Or and after you have submitted, got an accession number, you will go for the publication, and with the result that you have got from that study, you will create various illustrations, text, compile a manuscript and submit, right? So the others can also use the genome, right? So basically typically when you start, you have to get the genome approved by clearing all the ethical, legal and societal issues, because some genomes are highly protected genomes, endangered species, or you should not sacrifice an animal to do the genome sequencing, right? Even if that is desired or there is no option, you have to take proper approval before going for it, because it is like ethical issues and legal issues are involved here, right? And then basically you have to you may think like morphologically this is the genome of my interest, but you have to confirm that species that this is a genome using markers that we have discussed earlier, right? It could be mitochondrial marker, chloroplast marker in case of plants, or the nuclear gene which could act as markers, or typical traditional 18S, 16S, ITR agents which could be used as markers, right? Then you have to get sufficient amount of that tissue from that genome. Now what tissue will give you what information? or which is better to extract DNA is also challenging, because insects, which have a lot of chitin and other things. Similarly, plants which are full of phenols, terpenoids, and these kind of things, polysaccharides. So they may always interfere with the DNA extraction process and getting high quality pure DNA. So that is also a challenge. Then you should have some idea, even if, so you have basically extracted the DNA of good quality and high molecular weight if you want to go for the long read sequencing, because you want longer fragments, right, and longer much and get rid of the shorter fragments for the high long read, but shorter reads you can go for any, okay, because anyway in the library preparation step you will do size selection, right. Then you will assess the genome size. So either you will see that from the literature, some reports, or those information you will always be having from like your previous studies, or you have to do this ab initio from your study, from experimental or computational methods, right. Estimate the ploidy, again it could be done computationally or it could be known beforehand, right. Ploidy will really interfere when you are resolving the haplotypes. Genome size will tell you that how much you have to sequence, and that will also tell you that whether you are really capable of attempting a large genome, or you should start with a smaller genome of your interest and like that. Heterozygosity again is puts challenge while you are doing the assembly, and particularly in the case of de Bruijn graph and short read assembly. Okay, repeats region, again short reads, there are issues, but long read can cover these okay, and there sometimes like there could be symbionts which you will also think that you have extracted the eukaryotic DNA, but there could be some microbial species or micro eukaryotes which come along with your eukaryotic DNA, and you end up sequencing a large part of those also, and later on you have to resolve it by the computational means. Okay, then you have to based on genome size and all these parameters, you will do the planning of the sequencing, long read, short read, how much you need, synthetic long reads, or basically the 10x or link reads. Okay, then transcriptome sequencing, because transcriptome will tell you what were the portion of the expressed genes in that particular tissue, and will also help you when you are doing the gene prediction step, because or gene estimation step, correction of exon, intron, start end, because these are real evidences of the coding region that is present in the genome, which you have covered sufficiently in the transcriptome that you have sequenced. Okay, then basically these methods like Hi-C or RiboSeq, and these will help you to further sequence like linked leads, so that you can create a better resolution or higher resolution assembly, or highly contiguous assembly, which could be you can maybe call the chromosome level assembly. Then use the various assembly methods, including long read, short read, de Bruijn graph, or OLC method, right, or overlap layout consensus method, right. Resolve the haplotypes if it is like case of polyploids, and do the error correction polishing. Okay, then get the complete assembly, estimate again the genome size, repeats, and the various conserve elements or the genes from your assembly, right. Then based on physical mapping, Hi-C or optical mapping, you are going to resolve the chromosome level assembly, you want to gain that, right. So you will have a very good quality assembly by the long read, short read sequencing and assembly. Then using additional sequence that you did using the Hi-C sequencing, okay, you will merge them together, and based on these you will using the softwares you will try to create the chromosome level assembly. Okay, then you can do genetic linkage mapping if you desire. Okay, repeat identification, annotation of various genes, coding, non-coding, regulatory genes, regulatory RNA, etc. Okay, and then once you have done these, then you will go for more function analysis, where you will be looking at the typical features of the genome or characteristics, or connection between genotype, phenotype, and right, and evolution of certain characters in the genes in the genome of your interest. Okay, so basically at the start you will think of what sequencing technology you will use, long read and short read, create a hybrid, then think of the method that you are going to use for each and every step for Illumina sequencing or 10x Chromium or PacBio or a nanopore. Okay, then go for the assembly contiguity using scaffolding, various type of algorithm that you are going to use for assembly. Okay, separate as well as hybrid. Okay, and then in the end you will be basically correcting that assembly to an extent possible. So this is the path till here, which is going to tell you a good draft genome. Okay, but at the chromosome level, if to reach from here to the chromosome level, you have to include another step, that is the high C assembly or high C sequencing method, which we are going to discuss today. Okay, now for the high C, there are several requirements that it is not that you can do ab initio high C. So you all should always have have a good quality assembly in hand. Okay, so for example, based on N50, like you can decide, okay, which is one of the, if your N50 of the assembly is more than 1 MB, then it's a good starting assembly for the for going for high C. Okay, then if it is like 0.5 to 1 MB, again it is kind of fair. Okay, so basically this will tell that what is the length of 50% of your contigs are basically bigger than 1 MB. Okay, so that means that you already have a good quality assembly in hand. Okay, then coverage of your genome, which is like more than 90%, is good. Okay, but 80 to 90% genome also, if you have covered, is kind of fair. But if you are starting with these statistics, then basically you should not go for high C, because that means you have to again go for more rounds of sequencing or data generation, right? Improve your assembly, bring it to this level or this level. Okay, and then proceed for the high C assembly. Okay, so which is depicted here. Okay, then the Busco completion, again more than 90%, that means more than 90% repeats you could genes, single copy ortholog genes you could cover in your genome. So basically Busco is a very important matrix for that, and again more than 90% is better. And okay, so basically with this these three parameters, you will decide that you have good quality assembly, which you can proceed now for going for the chromosome level assembly using high C. Okay, so now high C is one of the methods to achieve the chromosome level assembly, which we are going to see today. Okay. Okay, so now for the chromosome scale genome assembly. Okay, you should have a prior assembly which should be done using the methods like Illumina, ONT or PacBio, Hi-Fi. Okay, which is a draft genome with high contiguity. That means your N50 was good, your Busco was good, and basically your genome completeness was also more than 90%, Busco 90%. Okay, N50 more than 1 MB or 500 KB to 1 GB, right? So basically these things are you should need before this. And then this draft genome is further scaffolded using the high C linking information that you are going to generate. Okay, additional sequencing that you are going to do using the high C library preparation approach. You are going again for the Illumina based short read sequencing, paired end sequencing, and then using that information to further do the scaffolding and construct a chromosome level genome assembly. Okay, so basically high C is a chromosome conformation capture technology. So which means that because chromatin in the cells or the tissue that you have taken is organized in the form of like loops, and basically has close proximity within the chromosomes, and that proximity means like sequences are close to each other, and that we can capture by high C. Okay, by fixing those in their relative conformation in a genome. Okay, so basically the assembled genome can be scaffolded using high C to construct a chromosome level assembly. So the contact frequency between pairs of gene loci or basically regions in your chromosome will help you to connect them better. Okay, and to construct the chromosome level assembly. So scaffolds are anchored, ordered and oriented based on the contact frequency. So okay, that means that you already had a good quality assembly which was in scaffolds, which were more than 1 MB in size, right? So now, but you don't know like which scaffold can, like there could be two large scaffolds, but you don't know this is coming from what chromosome, this is coming from what chromosome, whether they can really be connected this way or this way or this way, right? So that information, or basically you could connect multiple together to form even longer scaffold or ultimately reaching the chromosome. So that information we get from the high-C sequencing, right, and high-C based approach, right? And then okay, so these will help you to construct like chromosome length scaffolds, okay, which we just discussed. And there are softwares which can deal with the high-C data, take your previous assembly. You may also give information about the number of chromosomes you have, otherwise it can do on its own also, and then go for it. Okay, so okay, so basically high-C is trying to capture the information of the proximity of DNA sequences or DNA. Okay, so the DNA which is more closer to each other in the chromatin will be basically coming from the same chromosome. Most likely there could be some long distance also, but that can be resolved. Okay, and also that will tell that basically we will see in the future illustrations also that how the sequencing of these can help you to do long range organization of scaffolds. Okay, so before that, like basically it is recommended that your sample should be fresh. Okay, it is not that you will give a very old extracted DNA which may be subjected to degradation, or also high quality DNA is better. So practically your sample should be frozen in liquid nitrogen soon after you got the sample, and transferred in dry ice, and in between it should not like freeze thaw basically that means it was in the freezing condition, again you thawed it, bring to the room temperature or lower temperature, again you froze it. So that should be avoided because that will again cause damage to the DNA. Okay, and about 50 to 100 mg per experiment may be desired, and you should select a preferred tissue based on the organism that you are going to pick. Okay, like leaves or something for plant, or some heart or muscle tissue for human or like that. Basically these things you can know from the website or contacting the vendor. Okay, and basically then you even like cell cultures can also be done using high-C. Okay, so more details about this is available here. Okay, so now this is the overview of the high-C sequencing technology, which is trying to capture the long distance association. Okay, so in a cell, basically if you see like the DNA is organized in the form of chromatin, basically nucleosome and some free DNA and like that. Okay, nucleosome DNA is wrapped around the nucleosomes and form histones, and on the histones and basically form nucleosomes. Okay, so you want to capture the association or contact regions of the DNA. Okay, so basically in this picture you can see basically the region of DNA which was close to each other is fixed using formaldehyde. Okay, which helps to link the regions which are close to each other. Okay, then the cross-linked DNA is lysed by basically any a good restriction endonuclease or recommended restriction endonuclease like HIND3, which is basically a 4-base cutter. Okay, and then the regions are biotinylated. Okay, because you are going to do in the later a biotin streptavidin pull down technique. Okay, then basically these are ligated together, these regions. Okay, like this. Okay, and then the unligated regions or the regions that did not cross link are removed. Okay, then pure of these formaldehyde linked and basically biotinylated DNA is taken. Okay, so it will have two regions which were like red and black, for example here, which were distant in the chromosome. Okay, but basically showing a close proximity in the in the chromatin. Okay, then basically they are pulled down by using the biotin is pulled down with streptavidin, okay, and then these regions are amplified and basically read by sequencing, paired end sequencing, okay, to generate the high C contact maps to reveal the genome wide chromosomal assembly, okay. So this offers a very good method in which we can get the real info about how the chromosomes or the chromatin is organized in the cell, and their contact information will help to place the scaffolds that we get from the previous assembly in the relative order and connect in the form of a chromosome, okay. Like this, basically this is the cross-linked DNA, right, okay, right, and these two DNA was coming in close contact, which was cross-linked like this, right, and then this region was cut by hind 3, okay, and this part is removed, okay. So this is no more there, okay, and only the part which is linked stays, and it could be that you can get like unequal size part, or it is not two size equal because you are using a restriction enzyme, and depending upon the availability of site, like it's a 4 base cutter hind 3, so it will be like that, okay. So now you will fill the ends and mark with biotin, there could be blunt ends or ends also, you could make them even, right, and mark that with biotin. Now you will ligate these ends together, okay, make them sticky and connect together, okay, and then like in this way you can see here, right, okay. So now ligated, and now this biotin is pulled down using the streptavidin, right, and these regions are then sequenced by paired end sequencing of Illumina 150:2 sequencing, okay. Then after this you have got the sequence data. Now basically it is time to resolve the data and use the previous assembly data to achieve a kind of chromosomal level draft assembly. So various tools can be used for this, and PacBio, Hi-C, good quality, like basically a previous genome based on Hi-C, PacBio or ONT based plus short reads, error corrected, polished, is good for to start is a good input for the Hi-C. Along with the Hi-C data can be used together to get the final genome assemblies, okay. And ultimately this kind of quality genome quality can be used to achieve a telomere to telomere assembly, but basically, okay, so these three together can give you a very good draft quality. We discussed the matrices like how much parameter like how to estimate the quality so that it is good enough for the Hi-C, okay, followed by the Hi-C, and it can ultimately give you the like at this level you will get the chromosome level assembly, which can be used further to make the gapless telomere to telomere genome, okay. So basically the emergence of PacBio error free Hi-Fi is very important in this purpose recently, okay, plus ONT data because of its ready availability in the labs, and cheaper cost, and also easy to use. You don't need like a very big instrument with like in PacBio you need, okay, you can do this in the lab ONT. So that is also a very good way of achieving the long read, which can be used for the good quality draft genomes. Okay, so now Hi-C is will let you achieve the chromosomal level assembly. Now we should look at, when we are talking about what is a finished genome, like example of human genome, what we really achieved in a finished genome. So basically in human genome, first draft came in year 2000, okay. After that several drafts came, and recently we were all working with H38 assembly, okay, but still in that assembly like 8% of the genome was missing, telomere to telomere information was not there, okay, and which was achieved in the telomere to telomere assembly, which was made recently using this cell, okay. So basically it achieved the gapless assembly for all chromosomes except the Y chromosome, it corrected the errors in the previous assemblies. Okay, almost 200 million base pair sequence data was introduced in the previous assembly to fill up the gaps and the region that were unknown. Okay, and that included almost like 2000 gene predictions, of which like 99 were in the coding genes. That means we were really missing about 100 genes which could be of interest in health, in evolution, in various aspects that we were not knowing so far. Okay, which again like gives a like good input for the researchers who work on human genome and for further exploration, right. It included all the centromere satellite arrays, segmental duplications which are required to be covered. Okay, all the repetitive regions, short arms of all the 5 acrocentric chromosomes, and basically the regions which are important for structural and functional studies all were covered in the finished genome. Okay, so which is published in this article recently in the last year. Okay, so basically to achieve this, what they did was they extensively sequenced the CHM13 cell line. Okay, by using multiple technologies. Okay, they generated 30x PacBio data using HiFi. Okay, which is error free, or we see very less error data of long reads. Okay, 120x nanopore data, ultra long read sequencing, because basically both the technologies offer long read PacBio or ONT, and people based on the availability in their labs, they use both the technologies, right. And then basically 100x Illumina data, which is because already a version was available, aim was to fill up the gaps using long reads and also correct the errors in the previous assembly. So 100x more Illumina data was generated, and then 70x Illumina Hi-C data was generated to again look at the long distance chromatin contact information, right. Then BioNano for the optical maps of chromosomes, which is very important these days. Okay, and then basically they also did the single cell DNA template strand sequencing, StrandSeq, which is also a new technology to explore the other regions of these chromosomes which could not be covered by other methods, right. So ultimately if you compare the two assemblies of human, right, the latest one and the previous one, you can see that in the H38 it was 2.92 billion base pair genome, but in the current one it is 3.05. so basically about 4.5% difference was there in the two assemblies. Okay, unplaced bases were 0 in this assembly and the new one, whereas in the previous one it was 11 MB. Okay, then gaps were also reduced or removed. Basically number of contigs from 949 it became 24. That means earlier chromosomes was in 949 pieces, but now you have got the data into 24 chromosomes organized as per the chromosomes. Okay, and 50 increased to 154 million base pairs, and okay, in terms of genes, you can say earlier it was 60,090 genes, now it became 63,494 genes, of which almost 19,000 were protein coding. And basically you can see that there is a positive difference. That means there was an improvement, considerable improvement in the previous and the new assembly. Okay, that means a high quality draft versus a complete chromosome assembly. Okay, of which the new protein coding genes were also increased, right. Number of transcripts, isoforms were also detected, the new ones, right. And they could better resolve the segmental duplications, cover those regions, better mask the repeats, and identify all the different type of repeats in the new assembly. So basically now you have a chromosome in hand, or the genome in hand consisting of the 24 chromosomes, which you can say that basically that is the true reference of the human genome, and that can be used for the further studies. Okay, now that was the complete telomere assembly. But the draft genomes also are of good information and provide you a lot of data okay, and basically the start is always a draft genome which can later be completed, okay, by subsequent studies, and more and more availability of draft genomes will help you for understanding the medicinal, functional or typical characteristics of a genome, and also for the functional evolutionary analysis. Okay, so basically first of all it starts with you have to select what genome to sequence, because there are so many genomes available, millions. If you see around you, you will see so many genomes. Then you can always Google that, okay, what is sequence, what is not. Okay, if you are doing ab initio or random, or you're starting it, or it could be your lab interest, and you will check that, okay, this is sequenced or not. Okay, something that you are using as a model, or basically in your lab, or you think that that should be done. Okay, or if you're a student of ecology or evolution, then you may also find some genomes based on various objectives or hypotheses that you may have. So selecting the genome, then the hypothesis that why that genome is important and what you are going to find out of it. Okay, why that should be sequence, right? What properties you look for, what is your hypothesis about it. Okay, then the objectives that, okay, from this assembly I'm going to find just these, because these things will help you to also estimate the amount of sequencing and the analysis strategy, right? And what ultimately you are going to get out of it, whether you're going to publish it and share it with others, or give it as a template for further Hi-C or some other sequencing, right? Then you have to before starting you have to identify the methodology for covering all the objectives that you want to do. Okay, you can take pen and paper and see that these are the things to be done and who will do what. Okay, the analysis strategy, the algorithm, software. If you are already into genome sequencing and assembly and analysis, you will be having most of the pipelines or tools ready for. Okay, but otherwise if you are new, then you have to start from scratch, find out all the tools first, install them, see you have relevant set of infrastructure, computational and people, skilled people who can do the analysis. Okay, and before that, sequencing you may outsource, but analysis you may like to do yourself. Okay, even that you can also outsource, but ultimately the functional interpretation, or the hypothesis that you have and what you observed and expected, those things you have to do yourself to write a paper. Okay, then final outcome will be a publication or multiple publications or a patent, right? Depending upon the genome that you are doing. Okay, and these things will help as a reference of other studies, and you will be cited, lot of people will read this and also use that in their own work. Okay, and that is how things will move forward. So after you have achieved the genome assembly, the questions that are to be asked is like why that genome is like that, and the organism is like that, what changes happened in that organism so that it is different from its near species. Okay, that could be of interest, right? And how the what were the genomic clues you can get from the genome sequence analysis. So basically for this the orthology based analysis or orthologous analysis of orthologous groups that we covered earlier and also will be covering later will help you to do the evolutionary analysis. Okay, for example, the turmeric genome. Turmeric is one of the spice that is commonly used in our kitchens and also known in Ayurveda, and so much medicinal values, even cosmetics and so many skincare products and digestive products, pharmaceuticals, nutraceuticals, you find turmeric. But its genome was not sequenced till a few years ago, and then basically identification of these genomes that these could be sequenced is one of the things that you should keep looking for. Okay, so basically this family is Gingiberaceae, which also contains ginger. Okay, that was also recently sequenced. Okay, and containing about 1300 species. So these kind of information you will find from literature, and many people would have worked before this. So okay, so these are rhizomatous plants, aromatic, and okay, and so basically when you started, there were no species sequence, but now there are few species available from these. Okay, and it was known that some primary bioactives of this were curcuminoids, which are also responsible for the yellow color of turmeric. But how they are synthesized and their pathway, how those pathways evolved was not known. Okay, and basically turmeric plays a very good role as an antimicrobial, anti-cancer, antioxidant, anti-inflammatory. So we know these important uses of turmeric, but we never knew like what is the genome, and that is why producing the first high quality draft genome was important. Okay, so for which basically in terms of numbers, 82x or 94 GB, because the genome size was something like 1.33 billion base pairs, data of 10x genomics, and 47 GB or 41x nanopore genome data was sequenced. So generally like 25x or more nanopore data is like bare minimum that you should generate, and it was much more than that, almost 2 times of that. And the genome heterozygosity was also higher, so basically having more data is better, right? With a polyploidy case of triploid, which was identified and also proved computationally and experimentally. Okay, so estimated genome size came to be 1.15 GB, and along with this to resolve the genes and also to get the expression information of the genes coding part, to cover that in detail, 32 GB of RNA-seq or transcriptome sequencing also done. So when you extract the transcriptome, it is also contains the structural RNA, which is removed by ribodepletion, and only the mRNA or protein coding is taken forward for the RNA-seq. Okay, so basically using the leaf tissue of turmeric plant, 10x DNA was extracted, 10x genomics and Oxford nanopore was used. Supernova for the 10x reads, and the fly for Oxford nanopore reads, and genome polishing was done, scaffolding was done, okay, gap closing was done, and then the genes were identified with the maker pipeline, okay, giving the final gene set of 50,000 genes, and also RNA-seq as the empirical evidence for correcting the gene start end and the exon intron information. Okay, so this way basically any genome that you are going to attempt for a draft will happen. You may change things here, like the software that you will use, the contribution of nanopore or Illumina or PacBio, okay, at these levels, long shot, okay. The slight variation in the method of scaffolding or polishing, because so many new tools come, and also the previous tools improve, so you will always have options to change these steps, but overall methodology may look similar in the genome assembly things. Okay, then the assembled genome, the estimated was little higher, 1.12 GB, but assembled was 1 GB, containing 22,000 contigs. That means it was available in 22,000 pieces, and basically that means it is not assembled into chromosomes, and 50 was only 100 KB, okay, but 92% genes or BOOSCO completeness was there, okay. LAI score was more than 10, so basically it was a reference standard genome, okay, and basically almost 99% of 10x, 92% nanopore and 93% of RNA-seq reads could be mapped back to the original to the assembled genome. That means the genome that you assembled is basically covering all the information that you got from long read, short read, RNA-seq, and not much of that is actually left or you can say like left alone and not considered in your assembly that you got, okay. And 84,000 assembled transcripts were made, okay. The repeats were identified. Generally plants have like higher repeat, like 70% or more, okay. The 54,000 protein coding genes were identified, of which like almost 90% could be annotated. So basically with the chromosome level assembly or telomere to telomere, these gene numbers may change a little bit later, but to start with, if you are getting almost 90-95% complete genes, and that is a very good start for doing the functional comparative analysis. and also evolutionary analysis, and also for as an input for the other studies. Okay. Then identification of tRNAs and microRNAs are very important because many groups or researchers are involved in this research, and these kind of things have to be reported in the genome paper. Okay. Followed by the evolution analysis, because first of all you have to make a phylogenetic tree where you will accurately map your genome in the context of all the genome that are available close to your species of interest, okay, that you have sequenced. Okay. So there could be other phylogenetic trees also using small regions of the genome, but what you have achieved here is from a using most of the orthologs that you could find in the newly assembled genome. You have done this analysis, and this would give you more comprehensive and correct information. So it may agree with the previously available phylogenetic trees, or it will provide some information or some changes in the arrangement of species which could be reported. Okay. And then basically you can identify the factors that are important for it, such as stress tolerance, secondary metabolism. So you may have a hypothesis that okay, this is doing a lot of secondary metabolism, that is one thing, but from the genes that you have got and from the identifying the signatures of evolution, you will always get data that will tell you that okay, that these kind of things are really different in this genome, include, like for example here it was stress tolerance or secondary metabolism. Okay. Then like for a particular pathway such as curcumin in this case, you may like to go back and see that okay, first of all, what is the pathway, what genes are involved, how they are connected to each other, and then also to look at the origin of those genes, that whether this property originated in turmeric or it was already existing in some of the previous genomes, or there was some pre-indicators or pre-genes, or like slightly less evolved genes or something similar which is present in other genomes. So that thing you can get from here. For example, in this thing, the authors the genes that were interested in for a pathway, they looked at the EC numbers of that, identified the homologs from UniProt, okay, then aligned all those homologs together using cluster omega, okay, then they identified the functionally important residues which could be present at multiple locations in a sequence, okay, and if they are present in all the homologs and also okay, in the homolog of your interest, right. So basically those can be, that information can be used to select the hits, best hits which can be checked for this functionally important residues, and those can be taken as the enzyme of interest, and later on the remote homologs for these can be found by the HH blitz and other methods. Okay. So for example here this was the gene of your interest or enzyme of interest, so you could find a homolog in another curcuma species, right. Similarly here also for another gene you could find a homolog, but you could also see that basically something similar was also present in other species, and you could connect them using a phylogenetic tree, okay, because it is not that genes will originate in a single day, they must be having some undergoing evolution over a period of time, and then ultimately what you see today is the final expressed or the product that you are interested today, that enzyme may also be present in some different ways, or its ancestors in the other genomes. So that you can find in this kind of analysis, and then you can map all the evolutionary signatures in the pathways, like in the curcuminate biosynthesis pathway you can see that genes with unique substitution or genes showing MSA, right, multiple signs of evolution adaptations could be mapped, and this way you will know that what component at what proportion of a pathway is basically evolved and what steps are evolved, okay, which could again be a very informative evolutionary. Okay. And again gene family expression is another thing, because if you have higher copy number or of a gene, that basically that is also important evolutionary for various purposes, right. Similarly in the another pathway terpenoid backbone biosynthesis also you could map the things that were showing unique substitution or MSA genes or basically genes with no signature, right. So you could see that in this pathway, like most of the genes were having some signs of evolution. That means this pathway evolved in this species, okay. And ultimately you will make the final summary of the final outcomes, your interpretations, and submit that as a manuscript, okay. Like in this case, you've identified the stress tolerance, which was actually leading to the secondary metabolism, and it was producing a lot of secondary metabolites for combating the stress tolerance or achieving the stress tolerant. So it was doing it for its like own, okay, but not meant for medicinal properties. But when we started consuming this plant or its products, okay, those secondary metabolites that it was generating for itself to to tackle the stress issues, or basically for the stress tolerance, are having medicinal properties, and that we know today for in the case of turmeric. Similarly, for jamun, it is known that it is important for basically anti-diabetic, antimicrobial, antioxidant, so many other properties, and its genome was not known, and basically like almost all of its products are used for various purposes in Ayurveda and modern medicine, right. And it is from the largest genus. So basically these are the factors that will motivate you to go for its sequencing. And once you have achieved the sequencing by sufficient amount of nanopore and 10x genomics or Illumina data, okay, estimating the heterozygosity, polyploidy, it's a tetraploid genome, right, assembling the genome, right, with good quality N50 and LAI, right, and then basically identifying all the genes and then making the phylogenetic tree, right, placing that in the context of all the other species. And if for particular pathways like flavonoids, terpenoids, etc., which could be responsible for its medicinal properties, right, then identifying the signatures of evolution in those pathways, and ultimately linking all this information in the context of its medicinal properties, right. So this way for the genomes of your interest, you can follow these steps and do the evolutionary, functional, and the desired analysis, okay, and then make a final summary for that genome. Similar was the case for like ficus trees, either the people tree and burgat tree that were not known earlier, okay, sufficient amount of sequencing, okay, estimation of heterozygosity, estimation of genome size, right, a flow chart for the method that you have used, okay, Busco completeness more than 96%, sufficient amount of sequencing, okay. Then one PSMC plot, which basically this could also be one of the inclusion that shows that, okay, at this point there was a sudden decline in the population, right, where you can estimate that what point of time that happened, okay, evolution analysis, placing it in the context of others, making a phylogenetic tree of all these species of which are near to this or available, or their genomes are available, right. Doing the evolution analysis to see the genes for longevity. So basically burgat tree can live for like thousands of years, 2000 years, people know, right. Similarly for the people tree also, okay. So longevity genes or concept of longevity, or basically identification of signatures of longevity, could be one of the objectives, and you could find the functional insights into longevity and the genes that were responsible for the longevity in these genomes could be found by various evolution analysis, okay. So these are very subjective analysis. The question that you ask and the observation you make from the results that you get from the functional analysis. So I'll not go into details of these, but basically I'm just telling that how a genome story is built, and from collection of sample to sequencing to analysis to final publication, okay. Like disease resistance family could emerge as one of the factors that is conferring longevity to this genome, right, and you should present a detailed analysis of what genes were involved and in disease resistance and how they could be important, right, and ultimately make the story for the longevity concept in the case of say banyan or these ficus trees, okay. Then there was another genome that was there for a flower, okay. So basically you can say that the genome size was big, but still they could achieve a very high quantic N50. They could also do a chromosome level, arrangement of all the chromosomes and the regions that could were overlapping in those genomes, in those chromosomes. Okay, scaffold N50 was also high. They could segregate that into various 27 chromosomes. So this is a good example of a chromosome level assembly where the information was segregated into various chromosomes, the genome, the quantic and scaffold information. Okay, and also a high boost was achieved, right? So these kind of genomes will help you to take it further. This recently came and for the telomere to telomere assembly. Okay, so with this, basically what we covered in this lecture is we looked at the concept of assembly, what is a chromosomal level assembly, how we can achieve this using Hi-C, and what is a reference genome, what new you get in a reference genome which was absent in a draft genome. Taking example of a finished genome like human genome, and how the draft genome that you get from without going for telomere to telomere assembly, how those can be helpful for the functional and evolution analysis, and also acting as a starting point for further research. Okay, so this is good for today. Thank you.